same concept. They're just more pluses and minuses to look at. Yeah. Yeah. And I would totally do what I did. Circle the thing, whatever you need to do on your exam. Drawing arrows, bring colored pencils and pens. You guys do what you need to do to keep organized as you're working your way through these problems. That's totally fine. Don't feel like you can't write all over the exam. So long as your final answer is somewhat clear and squared off. Maybe a small apology if you write like Jason and it's like all over the page and fixing it. Yeah.
What's like the overall question? How many enzymes for mutants? What's the question?
The question is really asking how many genes were represented by your mutants, or how many of these. Because if you're kind of what you're wanting to do is right here. You want to generate mutants in all of these enzymes, several enzymes. You're not even told. So maybe if I told you there's 4 enzymes total. How many did you generate mutants for? You would say 2. 2 of the 4 total, yeah. There was, yeah. You couldn't go a whole lot further unless you were given more information about the whole path, the enzyme pathway or something like that. Yeah. So I'm going to start erasing this, but if you guys think of other questions about this complementation problem, just let me know. Okay. No other. So that was consulting my thing, because I always forget the numbers of the chapters in this book. That was chapter 7. Okay, so we're finished with chapter 7 now. That was your brief interlude of bacterial genetics. And we're actually going to move back to eukaryotic systems now. For those of you that hate prokaryotic systems and have prejudice against prokaryotes. Okay, so we're on chapter 5. Is even more so than some of the other exceptions that we talked about, is major exceptions to what Mendel predicted. Okay, so these are different ways that genes and alleles can be inherited, but really outside of what Mendel had predicted. It's actually a lot of really cool stuff in chapter 5. All of it is big topics of research ongoing right now. So I would say, I was sitting there when I was kind of thinking about this lecture and trying to count in my cohort, my peers in my PhD program, how many of them were working on epigenetics, especially, and a little bit on the imprinting side, which is another piece to this chapter. And it was like more than half of them. There's only 20 of us, but this is a huge hot topic, especially in human genetics right now, and biomedical kind of research and field. Which is somewhat ironic because I think they're more of the exception in terms of gene inheritance patterns. But these are really big areas that folks are currently studying. So I'll point out parts where we don't really know. I'll give you what we think and what the theory is and what the hypothesis is, but keep in mind that a lot of this stuff is still currently under investigation, and they're still trying to understand how this works. But we have a few of the rules, and those are included in your textbook. But for that brief introduction there, most of this stuff is less of that kind of heavy math problem-solving type of stuff, and this is a little more conceptual. So chapter 5 is going to tend to be more, can you understand the process and kind of think about what that might mean biologically, and there's less examples that I can pull from that are like, let's have charts of data, pluses and minuses, and count them up and calculate math frequencies and stuff, okay? So this is a little bit more thinking about the biology and conceptual. And this is the reason this whole chapter is really cool, is this is actually where the genotype, so if you have the genotype of an individual, it won't actually necessarily match the phenotype. So it may not directly even dictate or cause their phenotype. And this can be due to the timing of gene expression or gene inactivation. So we'll get to X silencing, if not tonight, next time. So these are cases where all of a sudden you have a given genotype, and this, as you can imagine, this totally screws up pedigree analysis. So you guys have been running through these pedigree analysis where you're assigning these genotypes. You're assuming that none of this weird epigenetic X silencing stuff is going on, and we haven't even looked at that kind of analysis, and you actually won't for file 311. So don't, you can kind of, yay, like sigh of relief. But that would totally, for a genetic counselor, really screw with their ability to make predictions based on a pedigree if the genotype of an individual is not even going to dictate necessarily the phenotype. Okay. And another mode of inheritance we'll talk about are those genes that aren't even found in the nucleus. Okay, so the genes themselves, the DNA sequence, aren't physically located within the nucleus. Yeah, so where are they? Mitochondria, right? So you have a whole genome that gets faithfully inherited actually through your mother. So we'll talk about mitochondrial genome inheritance. So this would be organized, and then what about in plants? So animals, we think of them having mitochondria genomes. Yeah, chloroplasts. So unfortunately we don't photosynthesize, but plants do. So they have chloroplasts. That's a whole separate genome that gets copied and inherited. So you are into plant genetics and crop genetics, which lots of folks are. It's another important area of research. They can also track the chloroplast genome inheritance patterns. So they can look for specific genes not found in the nucleus. Okay? So it gets a little complicated and crazy. Okay, so we're going to start, so that's kind of an introduction to this chapter, and we're going to start with looking at epigenetics. So, does anyone know what does epigenetics mean? What does that make you think of that word? Or does nobody have any?
Not related to the tags on the DNA.
Right. So a couple of things I heard are just totally perfect and accurate. Tags on the DNA, not related to actual, I didn't hear your other word, but sequence, nucleotide, bases of DNA. Okay. So this is where you're following a pattern of inheritance. That's doesn't actually involve the DNA sequences itself. So those base pairs, those nucleotides that we seem to be so concerned with, somewhat don't matter here, although I'll explain an instance where they do. So those base pairs or nucleotides, or however you want to think about it, that's not what you're tracking in terms of inheritance. Okay, so really this is DNA and actually chromosomal, so we'll think about chromatin, but both DNA and chromatin, so chromosomes themselves, these are modifications that occur on these two structures, basically. And we'll go into a little more detail later in the lecture about epigenetic inheritance patterns when we get to imprinting. So imprinting is an example or a type of epigenetic inheritance. These marks, so that's what the other suggestion was, one, that it doesn't involve the actual DNA, and the fact that epigenetics means tags or marks on the DNA. those tags or modifications occur really early during development, okay? So this would be at like oogenesis, spermogenesis. This is when gametes are being formed, those marks get put on the DNA. And they then control how that gene is expressed over the full lifetime of the individual. Okay, so that they're faithfully inherited in each daughter cell when there's a mitotic division. Okay? But they're not necessarily, these modifications are not necessarily permanent over generation to generation. They stay in place for a person's lifetime, but they get erased often and reestablished, again, like I was saying, in egg formation, sperm formation, gamete formation. So this is not a Mendelian inheritance pattern at all, right? We can't track them necessarily from generation to generation, right? Does that piece make sense in terms of they stay in place over your lifetime, but they will get erased between your generation and your children, or you and your parents, et cetera?
So you don't pass it on necessarily.
Not necessarily, yeah. And there's all these different ways that, and we'll talk about with imprinting how you can try to track some of that, but it's a little wonky, yeah. What are some examples of marks, or what do we modify? What kind of tags are added? Does anybody know?
Not, well, what you.
What proteins in chromosomes?
No,
That's a good idea. No, but
it has
there is a protein in chromosomes.
protein.
What kind of protein?
Sort of.
Found? Histones, right?
Yeah.
So yeah, so we can mark, you could mark either, so what I mean by DNA, you can also mark the histones, right? The proteins themselves. No, so it's good. So it's, you can either make marks on the protein or you can make marks on the nucleic acid. Okay? So you can, but what's one of the marks, or does nobody have? I'm just curious if you guys have heard of any. It's not like you need to know this answer unless you want to do this.
Methylation?
Yeah, methylation. So we'll do some Carl. So this idea of adding these methyl groups, okay? Methylation is definitely an epigenetic example, and you can methylate both histones and the nucleic acids themselves, so the DNA. What else besides methylation? This is, now you're really getting outside of.
Like RNA.
That, so, yeah, if you're thinking about like RNA binding to DNA or capsids, that's not really considered epigenetic. You might be thinking on a different line. Usually they're usually it's a small molecule, like carbon with three hydrogens. Another group that often gets, so there's a methylation, there's acetylation. So a lot of times acetyl groups actually get added onto, so this is like, did you listen in biochem or I don't know when you might have been exposed to this, but don't worry if you weren't. So methylation, CH3 groups are often added to histones and nucleic acids. And then acetylation, acetyl groups can also be added on. And the last really common one, and they're finding even more of these examples as this field gets bigger and bigger, but there's ubiquitin groups too. So there's this concept of ubiquitination. And what's ubiquitin? Does anyone know what kind of molecule? Anyone have to know?
Transport gene.
What is, so is it a nucleic, Jason?
Well,
Is it a,
it's isn't it an amino acid?
yeah, so it's a protein. Yeah, ubiquitin is a protein. So this, I'll let you give your little point. So these are small, we're just talking about little carbon, small chains here, but this is actually, it's small for protein, but it's a small protein that can get added on and hang off of the DNA and cause changes in the inheritance pattern and allele expression. Yeah.
And then usually isn't that put as a tag on proteins?
Ubiquitin can often be tagged on, so it would be the histones. Yeah. The histones in the chromatin would get ubiquitination. I can never say that word. It would get a ubiquitin group added onto them. Yeah. So that would be more of a chromosomal modification. Okay, so those are, you guys have now a few in your back pocket. You have a few examples of types of epigenetic marks. Okay. And we'll go into a little more detail. The imprinting part, that comes down to methylation, so methyl groups, and we'll talk about which nucleic acid, nucleotide that gets added onto and what that means for expression of that gene. Any questions about this little brief intro to epigenetic inheritance? All right, so I think we'll discuss next this concept of, I'm writing up here, dosage compensation. And really what this is this last item we have up here on our list, this X silencing. Okay. So you guys kind of already, I've asked you and you're totally on board with this concept, this idea that in a diploid organism, all your chromosomes are present as 2 copies, right? Except for what? Sex chromosomes, okay? In that case, female, so this would only be, in a diploid type of individual, for the most part, we're thinking of right now. The sex chromosomes are uneven, right? So females have 2 X's and males have 1. So on the X, you may not know this piece of information, but the X chromosome has about 1,000 genes. Okay, the Y I don't even remember. It's like 10 or 5. It's some crazy order of magnitude lower, and I should have looked it up, but we'll say hardly any. Okay. But either way, if you're talking about 1,000 genes, that seems fairly important, right, to our cell health, our cell mechanisms. We wouldn't want, so as a female, do you want those 1,000 genes? Is this double the expression? Do you have, is twice as good? Do you have more is better kind of thing? Or is it, so what's happening here, essentially, if you think about that? It seems really straightforward and easy to us. We always think of, okay, yeah, females have 2 X's, males XY, and that's normal and okay, and we walk around in our lives and are fine with that. But what does this mean? Does this mean that for men they have half the expression? No, we
don't women need more genes, especially for reproduction? There's so many. That's what
we
I
might
thought.
need more of other things, but no. So essentially, it doesn't. Yeah. So you're thinking kind of like, okay, well maybe that's a gender different. Maybe just in terms of biosynthesis of things. Generally speaking, maybe males and females they need that difference in expression. But it turns out no. You still want an even amount of expression. So just like your chromosome number 1, your chromosome number 2, chromosome number 3, etc. So we can think about abnormalities and understand that too in terms of trisomy 21. Okay, we know what that does. You have extra genetic material there. So if females had 2x, 2 times the expression here, that could certainly cause problems in species that have differentiated their sex chromosomes like this. Okay, some species of animals have the exact same setup of sex chromosomes between male and female. Okay, this is only specific actually to a few species, and I'll write some of them up here. But so it turns out that's not true. Men don't have, women don't have double the X, double the expression of those 1,000 genes. Men aren't at a half level. What ends up happening is you undergo, your chromosomes undergo dosage compensation. So this whole title of this section, dosage compensation, is talking about how different organisms deal with the fact that you have uneven numbers of sex chromosomes. Okay? So between sexes, there's an uneven number for that. Okay. So this concept of, I'll write this over here, by your dosage compensation, this takes on different forms depending on the species. Okay, so each animal is different. not all species are known, how they undergo dosage compensation at a molecular level, how that works isn't known. But there's quite a few that this has been studied in, especially from an evolutionary perspective. So the fact that these really different animals, these really different organisms have separately evolved independently to have some form of dosage compensation suggests what about dosage compensation? Yeah, it's pretty important, right? So if this is all evolved separately, this means it's critical for the survival of this species, okay? And those species that, again, have differentiated sex chromosomes, some have exactly equal, so it doesn't really matter. Okay, so if you think about we can make a little chart of the animal and then what the female genotype looks like and what the male genotype looks like for those sex chromosomes, and then how dosage compensation actually occurs, so how they handle this.
Like how it's shown.
It might make sense as I start to fill it out, but yeah, but how, whether one X is silenced or actually in some species what we'll see here is the X in males is upregulated. So either way we want this to equal. So I wrote up here half and then 2 times, but does it really matter? We could increase expression on this 1X for males, and then it will equal what females have expressing off of their 2X's, right? Or you could essentially get rid of one of the X's of females, and then you're still at an equal expression level as compared to a male. That's what I mean, yeah, by the how is kind of how do they deal with this dosage compensation issue? How do they deal with the fact that males and females look different on their sex chromosomes?
Okay.
So there's a whole class of marsupials. So the females are XX, males are XY. And the females undergo X silencing, okay? So this is similar to what we see in placental mammals like you guys, okay, so humans. The difference here is the females, okay, so they need to silence one of their X's, but they specifically, and I'll say for right now instead of silence, I'll say inactivate so that that's a clear concept here. So females inactivate, and they specifically inactivate the paternal X. So that's how they deal with the fact. And so then if they get rid of X that was inherited from dad, now they just have 1X with all those 1,000 genes or whatever marsupials have, and it's even to the expression level of the 1X in males. So each of their cells between male and female has an equal level of expression.
This was all marsupials?
All the marsupials that have been tested. So when I put these animals up on the board, these are the ones you have to go to the primary research to see, but all the marsupials that have been investigated. And so that's really similar to what I just said about placental mammal, where, again, the females inactivate an X. The difference here so 1X is inactivated. The difference is that it's random, okay, versus the paternal. And to know how that patterning works, so how, we're going to go into more detail about how in humans, how the X is inactivated, but that's a species-specific thing as well. The actual mechanism changes by species to species, but it's random in placental mammals. You have your fly, okay? You guys have been learning a lot about Drosophila, fruit fly. So sex chromosomes, female and male is XX, XY. The difference here is now all of a sudden males actually increase, they up the expression on their 1X to match that of the female flies. So males will increase expression of those genes found on their one X chromosome. So basically 2 times the amount that the female has on each one of her X's. The male flies will increase the expression on their one X. Your result amongst all these options that we're listing up here are essentially the same. You're just making them be equal in expression levels, but how they go about doing it has evolved in different ways. A couple more. So we have C. elegans, which is what? Does anyone remember? The nematode, right? The worm. Did you have a question in the back?
Maybe later.
Okay. So C. elegans is kind of weird. Okay, so C. elegans is XX and XO, and actually the XX isn't. You can put her under the female, but she's a hermaphrodite. And what the hermaphrodite does, okay, so the true male of C. elegans, the nematode worm, just has one X chromosome. It doesn't have the Y. So in this case, the hermaphrodite actually decreases expression on both X's at like a 50% level. It's really weird. This one's like the super weird one. Okay, so the hermaphrodite, which is the one with XX, okay, decreases expression on both of those X's. So overall, you have a 50% decrease compared to the XO male. Okay, one of my favorites is birds, of course. So does anybody know what the sex chromosomes for birds look like? Z's and W's, right? So what's the female? She's actually called the heterogametic sex. So she's the one that has 2 different types of sex chromosomes. She has a Z and a W. Okay, and the male is the one that has a dosage compensation issue, right? Because he has 2 Z chromosomes. So you can essentially think of Z's as somewhat to the X chromosome. Okay, in terms of the Z is the larger chromosome, it has many more genes on it. The W is a little tiny chromosome, there aren't that many genes located on it. Okay. These ones are really weird. We don't know as much about it. There seems to be some dosage compensation. All that's been done is that males and females definitely show differential gene expression on Z-linked genes. So when they look at a gene that's located on the Z between male and female, they're showing different levels of expression. So something's off here, but this is one of those current areas of research that's going on right now. Okay. Last one I wanted to leave you guys with, because this guy is just really cool. So this is the model for egg laying monitoring. What is that? Does anybody? Yeah. So platypus, which has a super bizarre genome. They just finished sequencing it, I think, within the last 5 years or so. So female has 10 X's. And the male has 5 X's and 5 Y's. And it turns out the males upregulate some of the genes. So this is again another one of those. A platypus is like the birds where it's still actively being researched, but what we see is that males, it looks like males are what we call upregulating, or turn up the expression, some of the genes. And those are just because they haven't been able to test every single gene quite yet. But I wanted to give you guys this example just because that's so crazy and bizarre looking. I mean, who
Some people said on
keeps
the
track
back.
of 10 sex chromosomes? That's absolutely bizarre. What kind of animal are you?
Does that mean that they have like 10 of each, or just sex?
So that's what for tonight. Don't forget to bring your exam. If you have that correction on problem 6, or if you just have a addition,